Okay,
Thank you,
so
Professor Sear. Thank you.
yeah, so good evening, and thank you for the kind introduction and this opportunity to talk to kidney patients and liver patients about a new inherited cause of liver and kidney disease that I discovered very recently. And so you might recognize and remember that Newcastle is a centre for excellence for inherited diseases, and we have the world-renowned Centre for Life, established by Professor Sir John Byrne a few years ago. And more recently, Newcastle has a new centre called the Centre for Rare Disease, where diseases that affect the liver, kidney, brain, and other organs are grouped together because of their rarity. And a common theme among a lot of these rare diseases is that it's usually an underlying genetic disorder. Okay, so for those of you that don't know the Centre for Life, it's this circular building near the central station, and you're welcome to come and say hi if you're passing. It's very handy for the city centre, and it has obviously the museum in there for the Science Museum. It has the fertility centre as part of the NHS, and it has laboratories, and this is where I do my work in the laboratory. So it's a great place to do science right in the city centre. So some of the stuff I'm going to be talking to you this evening is about genes and the collection of DNA in our body. The whole collection is called the genome. And there was great excitement back in the year 2000 because the first sort of prototype copy of the genome was published. And this had taken over 10 years and many millions of dollars in investment to establish the genetic code. You remember that DNA was discovered by Watson and Crick in 1953, but it took many years later for us to really understand how this DNA was behaving and how code led to life. And so along the way, along these 10 years, there was little breakthroughs and little discoveries, but it's only very recent history really that we can sequence someone's genome and begin to understand what it means. So in fact, this 10-year project called the Human Genome Project cost $3 billion. And just to think about the scale of things really, is that our genome contains 3 billion base pairs, and to store it on a computer, if you typed it out the code, it would take 3 gigabytes of memory. So that kind of tells you how big things are. Sorry, I didn't want to go forward there. So we're dealing with a large amount of code, and every time we sequence a human, we have to think about how much code is involved. But you might have heard the term junk DNA, and that's because when we sequence these 3 billion bases, about 90%, 97% of it, we don't really know what it does. And then in between are these coding regions, 2 or 3%, where we have begun to know what these things do. And so these coding regions are called genes. In between, we don't know their regulatory and structural regions, but the term junk DNA was coined and stuck a few years ago. And we are, as human beings, very close to other species. In fact, if you take a chimpanzee, we're 98% identical to a chimpanzee. But there's obviously enough variation in the genetic code to allow for huge differences between us and monkeys. So I mentioned the length of the code, but the big surprise when they sequenced the genome was the number of genes. They were expecting millions of genes, but the big surprise was that there was only about 23,000 genes. And so the whole of man or woman is encoded by quite a small number of genes comparative to the size of our genome. And even if you take a very simple organism like a worm has nearly just over 19,000 genes. So we're not much more advanced than worms in terms of our number of genes. And a fly, 13,000 genes, yeast, 6,000 genes, the tuberculous organism, 4,000 genes. So in levels of complexity, you know, we're of orders of 10 or 20 different rather than thousands of times different. And then to think about the number of cells in our body, and then that the number of genes and DNA. So there's 100 trillion cells in our body. And then if you think about it, there's 3 billion base pairs in each cell. That is an enormous amount of genetic material in our bodies. And so people have calculated various statistics around DNA. So if we unwound the DNA strands from just one single cell, it would stretch about 6 foot long, but only be 50 trillionths of an inch wide. Okay. And then this is my final sort of fact sheet on the genome. I don't want to bore you too much, but if all the DNA in your body was put end to end, it would reach the sun and back over 600 times. That's because of the sheer length of all these DNA strands and the number of cells in our body. And then the length of the code, a person typing 60 words a minute, which is pretty fast, I think, 8 hours a day, it would take them 50 years to type up the human genome. So that shows you the scale of how many letters in the code there are. And if these 3 billion letters in the human genome were stacked 1 millimeter apart, they would reach a height of 7,000 times the height of the Empire State Building. Okay, and this is what the code looks like. This is like a snapshot of some of it. And it's all A's, T's, C's, and G's, and these account for different bases of the DNA. This is what Watson and Crick discovered, these A's, T's, C's, and G's, and how they wound together in this double helix. And if you're someone like me, you really get a kick out of gazing at this stuff and looking for patterns and looking for changes in the DNA compared to normal. So as a real sort of different track now, I'm going to tell you about urine. So urine is my favorite sort of bodily fluid because it's usually sterile. It's tells us lots of information about the kidney. And the study of urine is like the oldest medical scientific test that there is. And people describe the study of urine on these ancient clay tablets going back thousands of years. And so the description and discovery of urine and what changes are in the urine was the basis of laboratory medicine as we know it today. And whenever a sort of test comes along, you get people that think they can interpret the test better than others. And so doctors back in the day, instead of carrying a stethoscope, used to carry these round bottom flasks and gaze at people's urine, and depending on the color and turbidity and various other parameters of the urine, they could make a diagnosis. And so these people were called piss prophets because they used the power of gazing at urine to make diagnoses. Fortunately, we have moved on a little bit since then. So you might recognize this entrance of the Freeman Hospital. This is round the back. And this is the new kidney unit and the dialysis unit, and it's tagged onto the cancer center as well. And this is where I see what I like to term my more interesting patients where I'm suspecting a genetic diagnosis and an inherited disease. And so I do a special clinic here with pediatricians and genetic doctors as well as myself, where we're looking for families with rare kidney diseases and looking to find a diagnosis for them and the most appropriate management and plan for them as they go forward. And so I get patients and families sent to me from across the whole of the north of the country to this clinic to try and help them. And these are some of the families that have come over the last years. And I like to think that I've helped some of these families to make a diagnosis and to help them get the best medical treatment. If you have a disease that doesn't have a name or a diagnosis, it's really hard to tap into the NHS resources because it's hard to describe what your child might need in terms of their medical input. As soon as you have a name for the condition they're suffering from, then it opens all sorts of doors and makes things still challenging, but a little bit easier. And of course, this clinic is an opportunity for my research where I'll take a urine sample from patients and blood samples for DNA along the way. And The set of diseases I'm most interested has a funny name. They're called ciliopathy syndromes. So these are disorders where the cilium, and I'll show you a picture of one of these in a minute, goes wrong. And the most common clinical manifestation of a cilia going wrong is cysts in the kidney. So here's a scan of the kidneys, and all these white blobs are cysts within the kidneys. They shouldn't be there. And what's quite interesting is whenever we see cysts in the kidneys, we sometimes see two things happen to the liver. Either the liver can become cystic as well, because if you think about the kidney, it's a bag of tubes filtering and making urine. The liver similarly is a bag of tubes making bile acids and cleaning the blood and making enzymes in a slightly different way. So it's not a surprise that kidney and liver disease sometimes goes together. So as well as cysts in the liver, sometimes we see a reaction in the liver that we call fibrosis, and this is the liver becoming unhappy. And occasionally we see other effects in the brain and the skeleton with these group of disorders called ciliopathies. But when I see a patient with a ciliopathy, I do take a urine sample and rush down to the Centre for Life with it, where we spin that sample down. And in any urine sample from anyone, you'll get a few hundred cells in a sample that have just been washed out naturally from the kidney. And we've learned to be able to persuade those cells to grow in a dish. And if we've got cells in a dish, then we can do lots of experiments on those cells. So this is what I've just told you, really. The kidney is a set of tubules. The functioning unit of the kidney is called a nephron, and we've got about 1 million nephrons in each kidney. And the nephron allows a filter to filter the blood, and the tubules to reclaim what's needed and get rid of what's not needed. And so this cartoon really shows a tube, and poking out into the center of the tube, the lumen, are these antennae, and these are the primary cilia. So these antennae tell the cells lots of things: what's left, what's right, what's top, what's bottom. They sense the flow of the urine, so they have a mechanosensor property, and they actually taste the urine, so they have what's called odorant receptors, parts of the cilium actually sensing the chemicals and other things in the urine to feed back to the body about the situation outside. So as I said, we take cells from the urine, try and grow them, and downstream from this, we can do really, really clever stuff. So we can do more genetic and genomic analyses on the cells from DNA. We can make RNA from the cells, which is the message taken from DNA, and do fancy things. We can look at the proteins from the cells. That's called proteomics. We can look at the cells under the microscope, under fluorescent stains, under high details such as the electron microscope. We can even persuade these cells to become stem cells, and then from stem cells make them into other organs or back to kidney if we want to. And the cells grow at the bottom of a dish in a single layer called 2D, or we can make them grow in 3D and start forming tubes. And so this becomes a very powerful research tool. And then just to reinstate that link with the kidney, this set of disorders that cause ciliopathies, all the genetic players have their protein products expressed in the primary cilium. So the most common inherited kidney disease there is worldwide is called polycystic kidney disease, and that's secondary to mutations in genes called PKD1 and PKD2. PKD1 encodes a protein called polycystin 1. Where does it end up? That protein ends up in the primary cilium. So if that protein goes wrong, you get kidney and liver cysts. And so there's been a paradigm now that states that really any gene that expresses its protein product in the primary cilium, if it goes wrong, will give cysts in the kidney and various other organ manifestations. Okay, this, if it plays right, is a picture of a cilium, and I want you to focus on the little things go up and down. So the cilium is a little bit like an escalator. So things move up to the tip and then back down. And I'll play it again, and I want you to focus on one blob. And what's fascinating is, a bit unlike an escalator, the velocity of things moving up is completely different to the velocity of things moving down. So what that means is the cilium has one set of apparatus to move things up to the top and a different set of apparatus to move them down. So what I'm trying to tell you from this information, this slide, is that it's highly complex and highly regulated, and the things going up and down are at a set velocity for a set reason, for a set sort of outputs. And so, like anything in biology and human nature, things, the more you start looking at them, the more complicated they get. Okay, so back to liver and kidney diseases. So I'm going to be now talking to you about these ciliopathies, and there are some known ciliopathies that affect the liver and kidney, and the best example is something called autosomal recessive polycystic kidney disease that actually gives you kidney cysts, but also congenital hepatic fibrosis, which is a severe condition affecting the liver. But, you know, I could be here talking to you about other liver and kidney disorders. Alpha-1 antitrypsin deficiency is one of those, polycystic liver disease that I've already mentioned, and Wilson's disease, which is a copper disorder. But, you know, these other subjects are for another time. But just to say that kidney and liver doctors do talk to each other, because we do look after patients that have, you know, shared diseases. And the pictures I've shown you before of cilia were in the kidney, but if you look in high magnification down a bile duct, you see exactly the same thing. So these primary cilia poking out into bile ducts within the liver, and again sensing the flow of bile and the movement of the bile and the chemical constituents of the bile. And so again, they're acting like an antenna, a sensory organelle to tell the cell information, what's going on from outside, and feeding it back to the cell. So very complicated structures. And this is an example of cysts affecting both the kidney and liver, which, as I said, often go together. So now I'm going to talk to you about this new disease that occurred, that I identified. And this was with the help of Linda, who entered the 100,000 Genomes Project, which is a way of us understanding more about the human genome and trying to sequence as many people with rare disease as we could. The 100,000 Genomes Project also recruited patients with cancers and patients with infectious diseases, but I was involved in the rare disease side of this. So Linda will tell you that she presented as a little girl with bleeding from her esophagus initially, and was diagnosed with liver cirrhosis that became more progressive as she got older and ended up with a liver transplant in her 40s. But alongside the liver, which was the main feature, she did, because she was having lots of scans on her tummy, she did have the finding of having kidney cysts as well, which is why she came to see me in one of my clinics. And so she did have these kidney cysts that were causing some kidney enlargement and a slight reduction in kidney function. But the 100,000 Genomes Project was very good at sequencing people, but not so good at finding new genes, because what they would do is sequence the whole genome, the 3 billion base pairs I've been talking about, and then they would put a mask over most of the data and just look at the known causes of cysts in the liver, or cysts in the kidney, or liver fibrosis, and say, okay, we've looked at what we know and we didn't see anything. And that's exactly what happened to Linda. And I think, you know, this was after a delay of many years of submitting the sample before she even heard back that we didn't see anything. Thank you very much. So to me, that represents a huge opportunity, that I've got a patient that I think has something wrong with their liver and kidneys, and I think they're connected because, as I've alluded to, these ciliopathy conditions often have kidney cysts as a major feature, and then other manifestations as other features. So I was convinced that Linda had something that was called a ciliopathy, and I wanted to explore it a bit more. So how do you find a new gene? So we have a list of things that we call candidate genes, and candidate genes are genes that look like they might be involved in the cilium, but no one has ever described a human being with that gene problem. So I'm always thinking about candidate genes, always thinking about how we could solve more and more patients with liver and kidney problems that aren't solved genetically at the moment. And so there are resources online to look for candidate genes, and there is a list of, it's called Cecilia Gold. There's a list of genes that they think this must be a ciliopathy gene, but we haven't quite proven it. And this gene in particular caught my eye because it was called TOLP3 because it seemed to have an important role in the cilia. And there was a mouse model, so a mouse that was genetically made to have this gene interrupted or deleted, and the mouse got cysts in the kidney, but there wasn't any human counterpart. So here is the gene inactivated in a mouse, and this is a normal kidney, and this is a massive cystic kidney. So this gene seems to be really important for the kidneys. They didn't comment on the liver in this patient. So long story short of probably many years of searching through the data from Linda, looking at the genetic code, looking at many candidate genes, we noticed that in the code there was a change in the gene TOLP3 that led to one amino acid being changed to another amino acid. So on the scale of things, this change is pretty minor. And if you saw it and said, well, no human has ever been seen with this change before, you might just discard it. But we got interested in it because, as I say, TOLP3 was a great candidate gene. And we had a hunch, really, that this was the explanation. So one patient in one family is never enough to convince anyone. And so we then began a UK-wide search, and then a European-wide search, and then a worldwide search for other patients that had the same liver and kidney problem, cysts in the kidney, liver fibrosis, and changes in this gene TOLP3. And so there was another UK family from Birmingham that was in the 100,000 Genomes Project as well. And then via collaborators in Germany and worldwide, we ended up with I think 15 patients in 8 different families, all with the same clinical phenotypes, so the liver and kidney problems, and all with changes in this gene TOLP3. And here is the gene written in the bottom, and the changes were all in different places. So there wasn't one mutational hotspot. Everyone had a different thing. And this is a diagram that one of my postdocs, Eric Ollinger, drew, and this is a timeline from birth to old age and what happens. And so these are the different organs affected. And so the kidney, when it goes black, that means kidney failure, and the liver, when it goes black, that means liver failure. And what you can see is that actually this sometimes is a childhood illness and sometimes is an adult onset illness, and there is a big spectrum. But the type of mutation that is identified means something. So if you have this change that we call a frame shift, which is a more severe genetic change, it seems to give an earlier onset, whereas if you have other changes, they lead to later adult onset degrees. The other thing that you'll notice about this slide is 3 patients actually had some heart involvement, and so we then carefully looked at the hearts of all these patients to make sure that we weren't missing any heart disease that had been overlooked. The kidney biopsies from some of the families allowed us to diagnose this thing called bridging fibrosis, which is quite a common finding within livers that are unhappy with portal hypertension and fibrosis. And the cysts, as I've mentioned, were of all sorts of, in the kidney, were all sorts of sizes and shapes, leading to polycystic kidney appearances. Okay. So what was known about this TOLP3 is that it allowed cargo to be delivered to the cilium. So remember that video I showed you of things whizzing up and down? Well, those because there's no protein production within the cilium itself, the proteins have to get delivered there. And so TOLP3, which is shown as this green blob in this diagram, binds to the plasma membrane via this protein PIP2, and then allows cargo to be delivered into the cilium. Within the cilium, then the cargo is released via TOLP3 because there's no PIP2 in the cilium. So that diagram encompasses several years' work by a group in the States that were working on this gene for a while. And what was really interesting about the mutation that we found in Linda was it affected this exact site where TOLP3 associated with this PIP2. And so you can imagine if there was a change in the protein structure at this exact point, then this interaction wouldn't work quite as well as it should. And that's exactly what we saw when we examined the protein structure secondary to this mutation. And so we took cells from Linda and took them to took a urine sample from Linda, took the urine to the laboratory, plated some cells out and got the cells to grow. And here we did some experiments looking at the protein levels of TOLP3 within the cilium that appeared to be normal. So that was interesting. But when we looked at proteins that were transported into the cilium by TOLP3, we showed that there was decrease in this protein GPR161, ARL13B, and IMPP5E. So these are all cargo proteins, and so we were able to prove really that the TOLP3 protein was there, but it wasn't doing its job in terms of delivering cargo into the cilium. And we did lots of other fancy experiments downstream from these cells, looking at the signaling pathways that were involved, and we got excited by some pathways called Wnt and TGF-beta, which were involved in fibrosis. And looking at these pathways allows us a glimpse of what the future could be, which drugs, which treatments might work to shut this kidney disease and liver disease off. And so it's very important that we get as much information as possible about this disease before we try and figure out how to cure it. And because cilia are involved in a process that causes, when they go wrong, causes extra aging of the cells, a process called DNA damage repair, we saw that in cells from Linda, there was a slight abnormality in this DNA damage repair, meaning, you know, the liver disease was probably a rapid, what we call a rapid aging of the liver. And again, this gives us insights into how we could switch this disease off in the future. So in summary, we found a new disease gene called TOLP3. It was a candidate disease gene, but if this gene goes wrong in humans, now we can predict that it causes liver and kidney and sometimes heart disease. And so this, because of our work using cells, we can now call it a new human ciliopathy syndrome, because the defect is directly due to different cargoes not reaching the cilium. And we've begun to try and understand which pathways downstream from this are causing the disease and think about treatments for this disease in the future. So clearly I didn't do all this work myself. There was my team in the laboratories in Newcastle, but also patients around the world, collaborators in different parts of the world as well. And, you know, none of this would be possible without patients giving urine samples and allowing this work to proceed. So I'll be delighted to interact with you and do some questions, but really thanks for inviting me, and thank you for your attention.
Excellent. So that's brilliant. Thank you very much, Prof. So hopefully we'll have some questions. I mean, there's some tremendous statistics, tremendous. You know, who thought about these mind-blowing facts and figures? I always try and have something ready in case someone tries to sell me, you know, something on the street, and I'll just give them some facts and figures, and they just look nonplussed and stop trying to sell me the whatever it is. I think we've got Yvonne would like to speak. You're on mute, Yvonne.
Yeah, thanks very much, John. Can you hear me okay? Yeah.
Yeah.
Thanks, Professor Stay. That was absolutely fascinating, really fascinating. And can I just say thanks to Linda as well for let, for letting us listen to, because it really did help sort of having the patient there. Do you know what I mean? It made it very real and how it actually works. But can I just ask a couple of things? Is it hereditary? And what you mentioned that obviously it affects the liver and the kidney. You mentioned the heart, but for instance, would cysts in ovaries, for instance, be something that would be looked at with that sort of potential?
Yeah, okay. So there's a few questions in there, but yeah, I'd also like to thank Linda, because Linda and I have obviously spoken a lot about this condition, and she's let me share pictures of her and things. And I think, you know, more so than kidney disease, liver disease is very stigmatizing. And Linda's told me about this, and that people, if you've got a liver problem, assume, you know, that you've been drinking or that alcohol's involved in some way. And so I think, you know, I'm very proud of the fact that Linda now is able to have a precise diagnosis, and it's nothing to do with alcohol. It's to do with genes. And so, to answer your question, yes, this is a genetic disorder. It's what we call a recessive disorder. So it wouldn't run in generations down the family. For Linda to get this, she's inherited one bad gene from her mom, her maternal side, and one bad gene from her father, and those two mistakes came together to give her that mutation. So in all the other families we saw the same pattern. So it was just, you know, came out of the blue by chance, because both parents happened to be carriers of this condition, and then it manifests, unfortunately, in the child. So we looked carefully at the heart. I'm still not convinced about the heart in some regard, but the three patients that had heart trouble had a kind of fibrosis of the heart, not any cysts within the heart. That fibrosis of the heart happened to people that were quite elderly as well, and so we've not really nailed that. We did, as well as thinking about the mouse, we did make a zebrafish. We haven't talked about zebrafish, but zebrafish are a model that we use to model disease. And the hearts were slightly abnormal in the zebrafish, and we're still trying to figure that out. So that's a work in progress. Regarding ovarian cysts, they're not related to kidney cysts at all. And so there are people walking around with ovary cysts and kidney cysts, but that's just two things, separate things happening in the same body. They're not related. Kidney cysts are really, really common. Ovarian cysts are quite common. And so people do have them both, but they're not associated directly. Maybe you could comment, Linda.
I was just going to say, anyway, thank you, Linda, for this, by the way, and for actually asking Professor Sayer to talk to us. That's brilliant, because we're full of liver disease specialists, and it's nice to have someone looking at it from a different angle. So yeah, would you like to comment, Linda?
Just to say that it is fantastic that I now have a diagnosis. As Professor Sayer said, you know, there is a lot of stigma, and I've always been very conscious of that. So now I can categorically say that it wasn't caused by me consuming excessive alcohol or either of my parents. But I just wondered, Professor Sayer, can you say how this discovery will actually play out? In other words, how will a patient be diagnosed with this new disease?
Yeah, good question. So the 100,000 Genomes Project was kind of like a political project in some ways. David Cameron, for all his faults, dreamt it up, and he said it should be possible on the NHS to have your genome sequenced. And when he said that, there wasn't the machines to do it, there wasn't the money to do it, it was just a complete political sort of statement. But actually, a bit like the moon landing in the States, we made it work. I don't know quite how, but you know, the government set up a separate company called Genomics England, and they bought the machines and they started sequencing people. And so that is why more and more people have got sequenced. And so now we have what's called the Genomic Medicine Centre, where now if I see a patient in the clinic and I think they've got something wrong with their genes, they might cysts in the kidney, funny liver, whatever, then it is now possible to have their whole genome sequenced. And you fill in the consent form saying, you know, I'm happy for you to look at my sequence. You fill in a clinical description form and fire it away, and it gets sent away sequenced in Cambridge or Leeds or Bristol, and then the data gets analyzed. So the sequencing is really easy, it takes about 7 days. The data analysis takes about 6 months to 12 months because as I've alluded to, there's a lot of data to look at. And so again, they put masks over most of the data because we don't simply understand it all. But so now when you've got a kidney or a liver problem, instead of ignoring this gene called TORP3, it would have previously been not looked at all. I've now, in making this discovery, anyone that has anything wrong with their liver or kidney, that gene will get looked at in the same way that the more common or well-known kidney and liver disorders get looked at. And so now, for the first time ever, we'll start making diagnoses of this condition because it's now, you know, that gene's on the map. It's there. We know that it's a player. Whereas before your discovery, Linda, you know, people weren't even looking at it because it wasn't known to cause any human disease. You know, they weren't bothered it causes problems in the mouse. But now, because it's a known human disease, we've got all these families from around the world. And since we published this paper, you know, there's been a flurry of other papers that thought, oh, I've got a patient that looks a bit like that, I'll sequence that gene. There's been a flurry of other patients that have been identified. And so, you know, it wasn't just us getting lucky, it was this genetic change was across the world if you look for it properly. So now it gives people a diagnosis. So you notice I'm being very careful not to say it gives people a treatment because we haven't figured that bit out yet. But you know, you've got to start somewhere. And so now we can say, okay, there's this condition, there's so many people all around the world with this condition. We need to figure out what's going on and see if we can treat this. So yeah, that's the idea.
Excellent. I think Yvonne's got a question then. Alf?
It was just on the back of that, Professor Say, from the answer you've just answered to Linda there, I was just wondering, does that mean that, for instance, me as a liver patient would be now getting a test regarding that? Are you saying that all patients, both kidney and liver, are all tested as a matter of course now?
Yeah, okay, so that's a good question. So I think if you've got kidney failure or got liver failure, no one knows exactly why, then that test is eligible. If you've had a biopsy of your kidney and liver and people have said, oh, it's this, then doing the genetics may be just confirmatory or maybe not necessary. But if you're one of these people that's just been told, well, hard luck, your liver's packed in, we don't know exactly why, that's exactly the sort of people that need to be having genetic tests because, you know, they're so powerful in discovering what's caused the underlying disease.
Yeah,
So your liver would have to be in the failure stage.
Well, it would have to have a diagnosis of some kind of liver disease, and then, you know, if that was thought to be possibly a genetic cause, then the genes would be sequenced. Yeah, I'm out of my comfort zone here because, you know, I'm not a liver doctor, but, you know, say for example your diagnosis was fatty liver disease, I don't think they would start sequencing everyone's genes for that. But if you had a more unusual diagnosis or something where they said, well, we don't quite know what it is, then that would be the time to look at the genes.
Is that okay, Yvonne?
Yeah, thank you. Thanks. John.
Right. Alf, you'll have to unmute.
Okay. Yeah. Really, I'm completely staggered by that talk. Fascinating talk. It seems to put certainly the details that you gave us initially put the Guinness Book of Records really in the shade. But what I would really, and having said that, it's also the personal effect with Linda being used then. You know, thank you very, very much for that. It really did put it into a context where you really began to put yourself in the position and you could start to see, you know, the real essence actually of the thing. And I was particularly interested in the science. I mean, this is science par excellence. When you look at how you can take cells, you can grow them on, you can do a whole series of experiments. That particular diagram of the psyllium showing the escalator effect was just remarkable. It does show at the micropsychological level, you know, how science has actually progressed. And then to take that and to start thinking about the pathways and how drugs can make you react. Everything which is good about science is actually there. Certainly genetics will come up one hell of a long way, you know, from Gregor Mendel indeed, and his experiments actually with peas. And I, for one, certainly will never ever forget TULP3. Thank you.
Okay, that's nice to hear.
And now Joan wants to say something.
Yeah, I was
Hey.
just going to say, we know someone who's had a diagnosis of cryptogenic liver disease, which has troubled her for many, many years. So would she be a candidate to have this, you know, investigation?
I would have thought so, because cryptogenic is just another word for I don't know, isn't it?
Yeah. Yeah, but it's, and it's exactly what you said. It's the fact that she doesn't know, and you know, she can't say to people, well, this is what I've got. And you know, when Linda said the confirmation that it wasn't alcohol, well, I would hope it wasn't at 11-year-old, but you know, it's well, you never know. But you know, it'll be the same for this person, where it's the stigma. Yeah. So it's a shame she's not here to have listened to it, because it was absolutely brilliant.
Yeah, great talk. Great talk.
Well, I think the recording will be available. I'm happy to answer anyone's questions if they want to email me as well. Yeah.
That's excellent. Is there any chance at all of a brief synopsis for the liver news? It doesn't have to be complex.
I think we've done one, haven't we, Linda?
Yeah.
Oh, of course you have. I'm thinking I'm miles away. This is because I'm on holiday. So I'm sorry about that.
Yeah, it wasn't that brief. It was quite long, but
Yes, been done. No,
yeah.
brilliant. Sorry about that. I'm always scratching for things for the liver news. I'm trying to dig up. Anyway, that's absolutely brilliant. Are there any more questions before we say good night to everyone?
Fabulous.
No? Oh, yes, Sean. Sean has a question.
I just wanted to say thank you very much to John and to Linda. You know, echoing what other people said. Amazing to hear the full story from both of your perspectives. Well, not the full story, but that overview of the story. Really very interesting. Thank you so much for sharing.
Lovely. Thanks for that, Sean. So thank you so much, Prof. Steer. Are you getting funding easily? Which is always a question I ask professors. Are you finding money flowing more easily now?
So, as I said, it's always a struggle, but Newcastle has made itself a name for rare disease, and you know, this Centre for Rare Disease within Newcastle University has recently got some big funding awards from the MRC, totaling about 3.5 million. So we're really pleased with that, and that will actually enable us to build on, you know, the small scale discoveries that people like me are making, and then take this out across the country.
Well, that's brilliant. If there's anything we can do to help, and if you need anything, please get in touch.
Okay, thank you.
Okay. That's it then, folks. I'm going to end the meeting for everyone. Thank you so much for your attendance, and thank you, Professor Steer.
Thank you.
Thank you.
Thank you. Thank you.
Bye-bye.
Bye now.